before they knew what DNA was, and they didn't even have a grasp of exactly what proteins were. The molecules of living things just hadn't really been well defined yet. But he discovered this idea that there was something inside living things that could be passed on from one generation to the next that controlled their traits. And so at that time, the gene was just meaning something, some factor inside an organism that gave it traits and could also pass those traits on from parent to offspring. And what he did is, what allowed him to discover the genes was working with pea plants. In the monastery that he worked at, they had a lot of gardens, and I guess he got permission from the father of the monastery to do some experiments. I might ask you some questions about these names, yeah. So he got some permission to do some experiments, and he would use different colored pea plants. The color is not very intense on this slide, but there was purple pea flowers and white pea flowers, and he would cross them with each other, meaning mate them with each other, and look at what the offspring were like. And let's see what the next slide shows. Here's something kind of interesting. In one of his test crosses, he made it a purple pea flower with a white pea flower, and all the offspring were completely purple. Now you might think at first, well, if I take purple paint and white paint, I get a very light pastel color purple. Does that happen? No, it was exactly the same shade as purple as the purple parent. So there was no, in this case, there was no blending of the trait. And the reason why I mention that is, before Mendel came along, people had a lot of incorrect ideas about how genetics worked. And one of the leading ideas at the time is the blending of traits, that if you have your dad's trait and your mom's trait, they thought you would be an exact mixture of the two. But this clearly showed that the traits were not mixing. One trait was essentially stronger than the other trait. But what was even more kind of interesting is, if you take these plants from the purple crossed with the white, if you take those offspring plants and mate them with each other, in the next generation, you get a few of the white ones popping up again. So it's like the white trait had somehow skipped a generation from this original crossing right here. And what's even more fascinating, you always get them in a certain numerical ratio. You get 3 out of 4 would be the purple color, and 1 out of 4 the white color. And so this was a completely unexpected result. Nobody knew that genes operated this way, and certainly nobody had done any sort of mathematical analysis of the way the traits went from one generation to the next. And Mendel had a mathematical background, and so he was not intimidated by analyzing things from a rigorous mathematical perspective. And anyway, by doing lots and lots of these mating experiments, he was able to come up with the rules of genetics, which are basically mathematical equations that show exactly why the white trait skipped a generation and why it showed up on average about 1 out of 4 of the offspring in this generation right here. And we're not going to get into his mathematical rules of genetics today. There's a genetics chapter coming down in a couple weeks from now. But the only point I'm making right now is he single-handedly discovered the science of genetics, but he had no idea what molecule was actually moving from parent to offspring. He knew its mathematical analysis, but nothing about the biochemistry of it. And he published his results, but was never able to say exactly biochemically what a gene was. Nobody knew. The next piece of the puzzle as to what exactly genes were made out of came about 20 years after that, after Mendel. This man right here is named Walther Flemming. He was a German scientist, and he was discovering, or not discovering cells were known by that point, but he was looking at cells under the microscope, and he was the first one to discover chromosomes. He saw these long thread-like things inside the nucleus of the cell, and he had no idea what their function was or really what molecule they were made out of, but he just said there's something in the nucleus of the cell called a chromosome that looks like a little piece of string. Well, about 20 years after Walther Flemming made that discovery, 2 other researchers, actually a whole bunch of other researchers, but these 2 guys in particular, Walter Sutton and, what was this guy's name? Theodore Boveri, were reviewing Fleming's results with the chromosomes, and they noticed that the number of chromosomes differed between reproductive cells like sperm cells and egg cells and all the other cells that were parts of your body. And that provided an important link because Sutton and Boveri and the others knew about Mendel's results, and some of Mendel's predictions about how genes worked would predict that you should get fewer genes present in the sperm and eggs, in the reproductive cells, than are present in the rest of your body. So these guys said, aha, there are fewer chromosomes in sperm and egg cells, and so that suggests that maybe the chromosomes hold the genes that Mendel discovered. This was the first association of a certain structure, a certain molecule within cells that were the genes. This is a diagram of the human life cycle, and the blue and the red things represent the chromosomes. And so these are, you can see the number of chromosomes inside normal body cells, but in the egg and the sperm, you can see half the number of chromosomes there. So I'm saying, as these guys said, that's exactly what Mendel said about the genes, half the number of genes in your reproductive cells. And so anyway, Sutton and Boveri were the first ones to say the chromosomes hold the genes. A few years later, there was a researcher named Thomas Morgan. He was an American fellow who was the first one to positively show that a certain gene, a certain trait, was associated with a certain chromosome. He was working with fruit flies. There's Thomas Morgan. He was working with fruit flies in his lab, and he noticed a mutation. Some of the fruit flies had white eyes, whereas normal fruit flies had red eyes. And by studying the chromosomes that were passed from one generation to the next, he found that white-eyed trait, that mutation, was always inherited when a certain chromosome was inherited. And so he said the genes are definitely associated with the chromosomes. That genes are somehow on chromosomes, so to speak. And so people became very interested in the chromosomes at that point to say, well, what about the chromosome is the actual gene? And let me flip briefly to the next block of text in your notes. So you can see chromosomes as thread-like structures if you look at a cell under a microscope, or at least during certain parts of the cell cycle, you can see that chromosomes look like little threads. A good diagram of a chromosome looks like this. The green dot that I'm showing in the center is called the centromere. And I think I mentioned this before. It's kind of a handle to move the chromosome around. During certain parts of the cell cycle, the cell needs to move chromosomes towards one end of the cell or another. That's what the centromere is, it's the handle. And despite the fact that they call it the centromere, it doesn't have to be in the exact center of the chromosome. Sometimes it's more towards one end than the other. But what we now know to be the genes part of the chromosome is the DNA, of course. So each chromosome is a long piece of double-stranded, double-helix DNA, in some cases millions of nucleotides long, but nevertheless one giant piece of double-helix DNA. And of course there are some proteins there also. There are these things called histone proteins, which act as spools for the double-stranded DNA to wrap up. And that's important because there's a lot of DNA inside each and every cell. Each tiny little cell in your body has about 2 meters worth of DNA if you stretched it all out. And so it takes a lot of wrapping up to take that much DNA and coil it up inside one tiny little cell. That's what the histone proteins are for. But what's kind of fascinating is after Morgan made his discovery that the genes were definitely on the chromosomes, almost everybody assumed it was those histone proteins and not the DNA that was the genetic material. And if you think about it, it kind of made sense because proteins are a lot more complex than DNA. DNA just has 4 monomers, the G's, A's, T's, and C's, whereas protein has 20 monomers, all the 20 different amino acids. And proteins did a lot of seemingly complex things. Proteins carry out different chemical reactions at their enzymes and their transport proteins and receptor proteins. Proteins just seemed a lot more, I don't know, complex and interesting, so people just assumed that they were the carriers of the genetic information. DNA seemed boring and didn't have any enzymatic activity by itself, and so people assumed it was just packing foam in the nucleus to surround and protect the histone proteins, which they thought were the genetic material. Most people thought that the proteins of the chromosomes were the genetic material, but the focus turned more towards the DNA thanks to some experiments by a fellow named Oswald Avery, who was following up on the work of another fellow named Griffith. And let's see, there's Griffith. Here's what Griffith's experiment showed. Griffith had some bacteria, and he had one strain of bacteria which was non-virulent, meaning it didn't cause any disease. It was kind of a weak bacteria. And he had another strain of bacteria which was very virulent. And so he assumed correctly that it was a genetic difference, that this had some sort of gene for being more of a disease-causing bacteria than this non-virulent one did. And what he did was he took the virulent strain and boiled it, sort of broke it apart into its bits. And when you boil a living thing, it comes apart into little bits. And he found out that he could take the non-living molecules, bits of that bacteria, and by transferring them to the other bacteria, that other bacteria would become virulent. And so some of these molecules must be the gene. Now, we now know, of course, that it was DNA bits that were being taken up by this other bacteria to transform it into a virulent one. But Griffith was not able to figure out what type of molecule was actually moving from here to here. He didn't know if it was a protein or a carbohydrate or a nucleotide or what. But he just showed that you could get transformation of the bacteria. And that's where this Oswald Avery fella came along. He essentially repeated Griffith's experiment of having molecules from one bacteria transform another bacteria, but he was able to purify some of the molecules that were transferring between the bacteria. And he showed the one that actually transforms the bacteria, gives it the new traits, was a DNA molecule, which is pretty conclusive evidence that it's the DNA and not the proteins that are carrying the genetic traits. The only problem was that at this point it was difficult to really fully purify DNA. And so even though what he showed that was causing the transformation was almost pure DNA, like 98% pure DNA, there were still a few proteins he couldn't purify away from it. And so a lot of the other scientists said, no, it's still those proteins that are causing the transformation. In other words, proteins are still the genes. The DNA you have there is just contamination. His work was important, but it didn't win over the entire scientific community that DNA was actually the genetic molecule. And then a few years later, another very different set of experiments were run that made people accept that a DNA was the genetic molecule. And these were called the Hershey-Chase experiments, which I think is kind of funny because it sounds like something you might make to make a chocolate milkshake or something like that. And they actually did use a blender in their experiment, which is kind of funnier still. This is Alfred Hershey and Martha Chase. And what they worked with is viruses to study DNA and genes. A virus is a very, very simple organism. It's so simple, in fact, that not all biologists consider them to be living things. All a virus is a very, very small chromosome with just a few genes on it, like maybe 5 or 6 or 10 genes. Compare that to the around 25,000 that human beings have. And surrounding the chromosome is a shell called the capsid, which is made out of proteins, and that just is a protective shell around the chromosome. And that's it. There are no receptors, there's no phospholipid bilayer, there are no metabolic pathways full of enzymes. It's just pretty much one chromosome and a protective protein coat around it. These viruses are such simple organisms that they can't even reproduce by themselves. Viruses can't divide the way living things do, or use glucose for energy the way living things do. The only way viruses reproduce, oh yeah, some of them have a little hook-like structure at the bottom to help them attach to cells, but that's just part of the capsid, it's proteins. Anyway, the only way viruses can reproduce is by tricking cells into reproducing them. A virus will land on a cell, inject its chromosome into the cell, and to the cell, this is just another chromosome. The cell doesn't know that this chromosome came from a virus, and so the cell starts expressing the genes that are on the viral chromosome. But of course, those genes are actually for the viral proteins, and so the cell starts making capsid proteins and other viral proteins, and eventually so many viruses get produced that they burst out of the cell, and that kills off the cell, and now you have copies of the virus coming out. So Hershey and Chase knew this about the viral life cycle, but what no one had researched yet was what comes inside the cell. Does the DNA chromosome come inside the cell, or does the protein capsid come inside the cell? And that was a very pertinent question, because remember, they were trying to figure out whether DNA or proteins were the genetic material. And so what Hershey and Chase did in some very elegant experiments was show that it was the DNA that goes inside the cell, and therefore that must be the genes of the virus and not the protein part of the virus. And I'll pick this up again on Wednesday. All right, I will see you in the laboratory for the exam.